Okay, in this video, we will be working on no, we will have a rundown on another flashcard Anki deck, Anki flashcard deck. This is basically gonna be about biochemistry. This is taken usually in the first quarter of grade 12 biology, um, as per the DepEd curriculum. Okay, we'll start. So, transport proteins, we have two types, channel and carrier. For channel proteins, they are hydrophilic, they love water, they channel water molecules. So, one example is aquaporins. They are used by passive transport. So, it may take longer for the transport of substances such as water molecules through channel proteins, but it doesn't require any ATP, doesn't require any energy, since again it is passive transport. So passive transport, no need for ATP. Carrier proteins, on the other hand, change shape and they transport glucose. So in order to transport glucose, you need carrier proteins which use active transport. So ATP or adenosine triphosphate, in other words, is needed. So aquaporins, what is their use? In kidney cell membrane, they enable the reclamation of more water from urine before excretion. So yeah, kidney cells. That's where aquaporins are useful. In closer to life terms, this privilege done by aquaporins keep you from urinating and drinking too much. To quantify, you wouldn't have to drink and urinate 180 liters a day. So this is true actually. It's a fact. Um, Without aquaporins, this is a tendency. So I don't think you do anything else in a single day, well, even at night, other than keeping needing to drink and urinate this amount every day. Anyways, molecules, so we have thermal energy, motion, diffusion. Diffusion is the movement of molecules of any substances down its concentration gradient from an area of higher concentration to an area of low concentration naturally. So there is no need for energy, it's spontaneous, and yeah, it is recognized that substances diffuse independently. How is this image relevant here? Not sure. Compositions of cell membrane. So we have lipids plus proteins plus carbohydrates. Basically everything in the list of macromolecules, well, biological molecules, except nucleic acids. For lipids, basically the cell membrane mostly has a specific type of lipids, that is phospholipids. They form a phospholipid bilayer. And proteins and lipids and carbohydrates, they are all arranged in a fluid mosaic model. I hope there's a picture that would illustrate this later in a different flashcard deck. Sorry, in a different flashcard. In the end, it's not the year. Wait, what? What the heck? Why is there a quote all of a sudden in this deck? We'll skip that. Okay, mitochondrion. This is the powerhouse for animal and plant cells. So in cellular respiration, this makes ATP molecules which provide energy for all cell activities. So the higher the energy, the higher the mitochondria count. So yeah. Again, ATP or adenosine triphosphate molecules provide energy for all cell activities, and they are synthesized by what? The mitochondria. So what you call the non-static sheets of molecules that are locked rigidly in place. They are held together by hydrophobic interactions. The cell membrane. So yes, this is what the phospholipid bilayer looks like. So here, this is the extracellular side or the exterior environment, and this one is with inside the cell, this portion, this region. In the middle, we have the cell membrane. So, yeah, we have polar heads and non-polar tails, which, as you can see here, the tails are in the center, so they are non-polar, and there's a reason why they are non-polar. And at the same time, the heads, which are seen outside, are polar. I just can't remember the exact reason right now. I need something that can trigger that memory again. Hopefully. So I believe because of being nonpolar, the hydrophobic interactions, they're responsible for holding the entire cell membrane together, as stated here. Yeah, I think that's already it. The tails are responsible for holding the entire structure together. Cell membrane, again, uh, in another perspective, it is a collage of proteins. They are embedded, it is embedded in fluid matrix of lipid bilayer. So there are two types of transmembrane proteins, integral and peripheral. For integral, they penetrate the hydrophobic interior of a lipid layer. Let's see. Penetrate. I think this is one example of an integral protein. They penetrate the interior. And another kind of protein basically just attaches to it, such as this. Let's see. Peripheral proteins do not embed in the lipid bilayer at all. Appendages loosely bound to the surface of the cell membrane. Okay. It's either this structure, but I think that's cholesterol, I'm not sure. Or this. I don't know why this picture doesn't have labels. But this is for carrier proteins, transporting from outside to inside and inside to outside. One, again, is for one uses passive transport, one uses active. One transports water, one transports glucose. Okay. Phospholipids form the main fabric of the cell membrane, and the proteins determine most of the membrane's function. What is the voltage across a membrane? Membrane potential. Okay, so we have the bilayer here, in the middle. The outside environment is over here. The cytoplasmic environment is here. Anions go up, cations go down. I can't elaborate more on that. Factors that affect membrane fluidity, we have three. First is temperature. So the higher temperature, the more fluid the membrane is. The lower temperature, the more viscous the membrane is. And number two, the type of the lipid membrane. The temperature at which a membrane solidifies also depends here. So how fluid it is. For an unsaturated hydrocarbon, like similar to the concept of saturated and unsaturated fats. For unsaturated hydrocarbons, from the term unsaturated, it's associated with the idea of fluid. So the membrane remains fluid in a low temperature. And for a saturated hydrocarbon, it becomes more viscous, not solid. Viscous in a higher temperature. And number three, presence of cholesterol. There is a fluidity buffer for the membrane. Cholesterol is a fluidity buffer. It resists changes in membrane fluidity that is affected by temperature. So, in a high temperature, so basically, the presence of cholesterol it causes the opposite. It causes a reverse in what usually happens in the application of temperature. So here, initially for the first factor, if there's a high temperature, the membrane is supposed to be more fluid. But here, number 3, in a higher temperature, it's the opposite. It doesn't become more fluid. It becomes more viscous or solid. So the same goes for a lower temperature, but cholesterol is involved. So the membrane becomes more fluid, not more viscous. Okay? Dialectics. This is not science at all. This is supposed to be a different subject. I think it's philosophy. I don't know why it ended up in this deck. Okay. Cytoskeleton. This is the blank of cell, the framework of a cell. It is made out of 2 things, microfilaments and microtubules. Microfilaments are made of protein, and microtubules are thin hollow tubes. Where's your philosophy? Okay. What is true about diffusion? Basically, molecules move blank their concentration gradient down their concentration gradient. Substances diffuse independently of other substances. So one question, where okay, I don't get this question. Let me think. Okay, so in a scenario where there is an imbalance in concentration, diffusion happens. Diffusion of substances down its concentration gradient. One example of diffusion is the uptake of oxygen by cellular respiration, and that oxygen diffuses into the cell across a plasma membrane. Membrane carbohydrates. We have glycolipids and glycoproteins. For glycolipids, we have some are covalently bonded to the lipids. For glycoproteins, most are covalently bonded to the proteins. Diverse membrane carbohydrates. They serve as markers that distinguish one cell from another. One example is, one application is ABO blood type. So each blood type contains different glycoproteins on the red blood cell surface. So glucose bonding to the proteins and lipids will always be covalent. Okay, we've seen this figure earlier. This is the channel protein. Integral protein, peripheral. Yeah. I think my guess was correct. Cholesterol is the thing that you see within the it's basically embedded inside, like a blackhead. And glycolipids just attach. I think in a way it dangles. Carbohydrates over there. Glycoproteins. I'm not sure what this is for. The globular protein. Was this mentioned earlier? I'm not sure. I don't recall. So skip that. What is this? So many philosophy questions here. What the... Okay. Endocytosis. This is the process in which substances are brought into the cell. So yeah, from outside to inside. A small area of membrane sinks inward, the vesicle formed containing material from the outside. So there are 3 types of endocytosis. We have phagocytosis, pinocytosis, and receptor-mediated endocytosis. For the first type, phagocytosis is in other words called cell-eating. So if the intake involves a large substance, then that's phagocytosis. For pinocytosis, cell drinking, the substance that is being consumed from outside to inside uh, is small, basically. For receptor-mediated endocytosis, um, usually it's associated with the idea of cholesterol. So it is the entry of cholesterol inside the cell. LDL, I know this stands for something. Uh, Basically, they act as ligands, molecules that bind to LDL receptors. Let me just determine the meaning of LDL. Okay. Low density lipoproteins. So, yeah. I know this is not healthy either. It's not a good kind of cholesterol. So, in particular, we have familial hypercholesterolemia. This is a genetic disorder. So, there is an increase in cholesterol level in blood since LDL receptor proteins are either defective or missing. So, it's important for these receptor proteins to be normal. Otherwise, you'd have a higher risk of atherosclerosis even if you don't live an unhealthy lifestyle. You don't eat fast food, but you'd still get atherosclerosis because of this. It's already stated here. Low density lipoproteins. These are bad cholesterols. I just said that. Chloroplast. This is where photosynthesis happens. So it's green because of the pigment chlorophyll. And when we say photoautotrophic, basically it captures sunlight for energy. Stroma, thylakoid, inner membrane, outer membrane. I already forgot, honestly, the functions of thylakoid and stroma. I might be able to remember it later. Can't guarantee anything. So, cell-to-cell recognition. So this is the cell's ability to distinguish one type of neighboring cell from another. So, for example, sorting of cells into tissues and organs in embryo. And the rejection of foreign cells by the immune system. That's it. Okay, easy question. Which of the following environments matches the correct animal or red blood cell condition? This card is a little broken, actually. The question doesn't make sense with what's provided. After pressing the spacebar, you already see everything. Anyways, this has already been mentioned earlier. I won't discuss that anymore. For a cell without a cell wall, basically plant and human cells, they cannot tolerate excessive water loss or intake. They cannot tolerate excessive solute loss or intake. Basically, they cannot live in extreme environments. They can only live in an isotonic environment. Okay, recall. The tonicity is basically the ability of surrounding solution to cause a cell to either gain or lose water. So in a hypertonic solution, higher solute, lower water. So that's the proportion. In an animal cell, it shrivels. For a plant cell, it gets plasmolized. Okay, too much salt, basically, too much solute. Sodium potassium pump. This is the pump that transports sodium ions out and brings in potassium ions into the cell. So there is basically a transfer of one charge, positive one charge from cytoplasm, that's from the inside, to the outside for voltage. It helps maintain a high concentration of sodium ions outside the cell and a high concentration of potassium ions inside the cell. So how is this kind of pump important? So it's critical for the electrical signaling of neurons that's in the brain. how fast your brain responds to certain stimuli, something like that. On the contrary, we have another kind of pump. This is the proton pump. This is the electrogenic pump in plants, fungi, and bacteria. So there is a transfer of a positive charge from the cytoplasm to the extracellular fluid, so from inside to outside. How is this important? This is used in ATP synthesis during cellular respiration. Again, ATP is synthesized by the mitochondria. So the mitochondria is involved in cellular respiration. So for the sodium potassium pump, transfer of one charge from inside all the way to the outside. So sodium ions delivered out, as you can see here, and potassium ions are brought in. There, so more potassium ions. So basically, it's just to maintain a balance of sodium and potassium ions. Okay. Proteins on the surface of a cell help outside agents invade the cell. So one example, one application is that protein surface cells enable HIV to infect cells. So this is usually the case for most people, but not all people have protein surface cells that cause them to have HIV. So in a way, they are immune to HIV, even if they get exposed to surefire ways that can cause them to have HIV. But in the end, they don't, because they don't have the specific protein surface cell that gives them, bridges the HIV virus to them, to their system. So researchers are developing drugs that can block the cell surface receptors involved in HIV infection. So from the observation that certain people don't have this kind of protein surface cell, scientists want to see if we could actually get rid of that kind of protein surface cell for those who can actually be infected by HIV in the first place. So yeah. The goal is to mask CCR5 and block HIV entry without affecting the CD4 receptor. So masking the CCR5 cannot be without affecting the CD4 receptor. That's the tendency. So yeah, we are yet to find newer news about this thing. Okay. Temperature. A membrane remains fluid as temperature decreases until phospholipids settle into a closely packed arrangement. Then, the membrane solidifies when the temperature decreases, in other words, cooling. Okay. So again, tonicity, ability of a surrounding solution to cause a cell to gain or lose water. In an isotonic solution, water diffuses across the membrane, but at the same weight in both directions. So in a certain sense, there is a balance in the proportion of solute and solvent. For animal cells, they are normal. For a plant cell, they are flaccid. Basically, in other words, clumsy normal. So there is no net flow of water. Okay. Yep. We're finished there. Okay. One of the organelles that are present in a cell usually have the Golgi apparatus. Although I think in bacteria, they don't have this. I think. Because they lack endomembrane organelles. they compensate in a certain way. I guess through their plasma membrane, it has more functions than human, animal, plant cells, and fungi, I think. Anyways, Golgi apparatus, in other words, the Golgi body or Golgi complex, they are responsible for receiving proteins and other materials from the ER in small vesicles. What is the ER? It's the endoplasmic reticulum. They customize proteins into forms by folding or by adding other materials onto them like lipids or carbohydrates. So this is the Golgi body. And yeah, rough ER is over here. Nucleus, cytoplasm, the entire thing. Mitochondria over there. I think these are lysosomes, I'm not sure. How do Golgi bodies modify the proteins into forms that can be used by the cell? By folding it into usable shapes and by adding other materials such as lipids and carbohydrates. So in a certain way, it's like a factory for protein folding. So, let's talk about active transport. What is true? Active transport moves solutes against their concentration gradients. So that's true. Transport protein used in active transport change its shape in a manner to translocate a solute across the membrane. I think one solute example is sugar, especially that active transport is involved here. So the transport of sugar requires ATP. A transport protein transports sugar from inside to outside and vice versa with the use of ATP. Active transport does not use channel proteins. Instead, they use carrier proteins, which transport complex sugars. And ATP powers active transport by transferring its terminal phosphate group directly to the transport protein. So what is false here? Well, this is false because, again, active transport does not use channel proteins, only carrier. Okay? Every other statement here is true, and it's already typed here. Types of cell wall materials. For a typical plant cell, we have cellulose. For chitin, that's the cell wall of fungi are made of chitin. I'm not even gonna bother saying this. But for certain prokaryotes, their cell walls are composed of peptidoglycan. When cells are immersed in a hypotonic solution, basically the walls help maintain cell water balance. So the hypotonic solution is like rainwater. There's not enough solute, there's too much water, basically. Okay, that's already finished. Cytoplasm. This is where all cell organelles float around. Ribosomes, on the other hand, we have two kinds. Free ribosomes, they are located in the cytoplasm. They produce proteins such as peripheral membrane types. And some others work in the ER. So, yeah. Facilitated diffusion, considered passive transport. So the solute is passing down its concentration gradient. The process of facilitated diffusion requires no energy. It doesn't require any ATP. Diffusion is known as the net movement of molecules from an environment of high concentration to an environment of low concentration. So what are the three types of diffusion? According to this, according to someone, we have three types: simple, osmosis, and facilitated. For for simple diffusion, from high to low concentration, uh, transpiration in plants, gaseous exchange during photosynthesis. For osmosis, the second type, the particles moving are water molecules. They move from a region of low solute concentration to a region of high solute concentration across a semi-permeable membrane. So, yeah, if you remember the U-shaped illustration, that's exactly what osmosis is. So, for example, if water and salt solution are separated by a semi-permeable membrane, then the tendency is water molecules flow to the region of salt solution as it has higher salt content. To where there is a higher solute content, that's where water would go. And number three, facilitated diffusion is the movement of hydrophilic molecules or ions across the plasma membrane. The difference is the diffusion is facilitated by transport proteins. For example, carrier or pore proteins. All of these three types of diffusion are forms of passive transport. So they don't require energy. Okay. So what is true about the sodium-potassium pump? So these are all true statements. The pump exchanges sodium ions for potassium ions across plasma membrane of animal cells. So basically you release sodium ions from inside to outside, and you bring in exchange um, potassium ions from the outside, bring it inside. So for each crack of the pump, there is a net transfer of 1 plus charge from the cytoplasm to the extracellular fluid, which stores energy as voltage. So this is considered the major electrogenic pump of animal cells. And basically, to quantify, 3 sodium ions are pumped out of the cell for every 2 potassium ions that are pumped into the cell. Okay? Fluidity of membranes. We have lateral movement, they shift laterally, and flip-flopping, they flip-flop transversely. Membrane proteins move more slowly. Some drift, some move directly along skeletal fibers due to the help of motor proteins. Motor proteins are connected to the membrane's cytoplasmic region. Many other proteins are held immobile by their attachment to the cytoskeleton or the extracellular matrix. In hypotonic or hypertonic environments, animals that lack rigid cell wall must have adaptations for osmoregulation. So what is osmoregulation? This is the control of solute concentrations in a water balance. So what are examples for adaptations of osmoregulation? Pico datum living in pan water. Pan water is hypotonic. The animal cell is supposed to lie in a hypotonic environment. But for this particular species, the Pico datum, it does have a plasma membrane that is much less permeable to water. So, in a certain sense, its membrane acts more like a cell wall in a plant cell. So, even if technically Pico datum, a Pico datum has, technically it's an animal cell, it doesn't burst or lyse. So, yeah. Another factor that contributes to the adaptation of Pico datum in surviving a hypotonic environment It does have a contractile vacuole that forces water out of the cell as it enters by osmosis. So even if there's a tendency for water to um, eventually accumulate from inside, there is a vacuole from within that brings out water. It basically expels water from inside. So yeah, that's it. Is fluidify even a word? Ah, wait. No, I don't think so. Maybe this is just um I think I just made this word up. But basically, membranes must be fluid in order to work properly. So when membranes solidify, permeability changes and proteins become inactive, specifically enzymatic proteins. When membranes fluidify, uh, they impair or they cannot support protein functions either. So therefore, extreme environments pose a challenge for life. Okay? Chromatin. It's detangled and spread out form of DNA inside a nuclear membrane. So this is a DNA helix. Chromatin is over here. Chromosome is the entire thing, I believe. Okay, what is ATP? It supplies energy for active transport, and its terminal phosphate group is directed to the transport protein. Again, transport protein, it is used by ATP so that substances or molecules such as glucose could be transported from inside to outside and vice versa. Okay. Nonpolar molecules are hydrophobic. So there is semi-permeability in the membrane. So molecules cannot just easily cross through, or they cannot pass through easily. So nonpolar molecules are hydrophobic. They pass through the membrane easily and without the aid of membrane proteins. So what are examples? Carbon dioxide and oxygen. So when we breathe, um, they don't need the help of membrane proteins in order to pass through the membrane. For polar molecules, on the other hand, some examples of which are water, sugar, and sodium, they pass slowly. They need membrane proteins to pass through. Again, why is sodium here? In a sodium-potassium pump, again, 3 sodium ions are pumped from inside to outside. So technically, it's also here. Sugar is taken in, so from outside to inside. Water as well. Vacuoles. Vacuoles are sac-like structures that store different materials. So, for example, in plant cells, these store water. For plant cells, vacuoles store water, and starch is plant stored sugar. Organelle, in other words, little organ. It's a specialized part of a cell that has a unique job. So we have the nucleus, nucleolus, ribosomes, rough ER, smooth ER, microtubule, cell, centrioles, Golgi body, lysosomes, the garbage collectors, mitochondrion, powerhouse. Nucleus, nucleolus, well, at least the nucleus is basically contains the genetic information. Ribosomes, they make the protein, I think. Yeah. Some are free, floating in the cytoplasm. Some produce proteins in the rough ER. In the smooth endoplasmic reticulum, on the other hand, I think this is where lipids are manufactured. So if rough ER is responsible for the synthesis of proteins, adding carbs and other accessories, in a way, the smooth ER is responsible for making lipids. The cell wall is outside the plant membrane, and it shapes, supports, and protects the plant cell. Okay. Again, the cell wall of a plant cell is made out of cellulose. Okay, all cells have three things in common regardless of type: cell membrane, cytoplasm, and DNA. So according to the CK12 Foundation, there are four things that all cells have: cell or plasma membrane, cytoplasm, ribosomes, and genetic material, whether it's DNA, RNA, or genes. So yeah, conflicting information. I've also noted that before when I was making this deck. Okay. Smallest living units of an organism, the cell. Yep. Voltage. Electrical potential energy, separation of opposite charges. So in the outside, or in other words, the extracellular side, there is a positive charge. This is the phospholipid bilayer, the plasma membrane, in other words. Within the cell, there is a negative charge. Various proteins are attached to the lipid bilayer. So it's seen in this model, the fluid mosaic model. ATP is, in other words, adenosine triphosphate. Okay, so there's a phosphate group, a ribose, and an adenine, or a nitrogenous base. Adenosine triphosphate, from the prefix tri, there are three phosphate groups. Lysosome, in other words, garbage collectors. They take in damaged or worn out cells, and its enzymes break down the cellular debris. I wonder when is this gonna be finished. I'm getting tired. Okay, passive transport. Diffusion of a substance across a biological membrane without energy. So the concentration gradient represents potential energy, and this drives diffusion. So for nonpolar molecules, such as oxygen and carbon dioxide, they can diffuse passively. They can cross the lipid bilayer without any issue. But for polar molecules, such as sodium, water, and sugar, or glucose, maybe water, for example, not sugar. They can diffuse passively. They cross by transport protein. Facilitated diffusion. That's what it's called for the diffusion of water molecules. Because again, to reiterate, water is polar. So what is facilitated diffusion? It's the passive movement of molecules with the help of transport proteins. Most transport proteins are very specific. That's something you'd want to note. What are the requirements for facilitated diffusion to happen? There is a transport protein, there's a cell membrane, and there is a concentration gradient. Without any of these, Facilitated diffusion will not happen. Nucleolus. This is where ribosomes are made. Really? Ah, yes. So, ribosomes make proteins, but ribosomes are made in the nucleolus. So, when the cell is ready to divide, the DNA condenses into chromosomes. Eventually, they would undergo mitosis or meiosis. Ribosomes synthesize proteins. I've already said that before. Soluble cytoplasmic proteins, peripheral membrane, mitochondrial, chloroplast, peroxisomal, and nuclear. Those are the kinds of proteins produced by ribosomes. Exocytosis. This is the process by which molecules are released to the outside of the cell. So there is a fusion of vesicle membranes and cell membrane. What are real-life examples? Pancreatic cells secreting insulin. Neurons, basically nerve cells, releasing dopamine. One thing to note is that there are two types of vesicle transport, exocytosis and endocytosis. So yeah, endocytosis is the opposite. It's not the release to the outside of the cell, it's the intake to the inside of the cell. Okay, all cells have three things in common regardless of type: cell membrane, cytoplasm, DNA, maybe even ribosomes. But that depends. But then to be safe, we have these three. So the cell membrane separates the inside cell from the environment. Cytoplasm, where all organelles stay afloat. DNA contains the genetic material. Nucleus is the control center of the cell, in other words, headquarters. It contains DNA or the genetic material. What is the DNA? It dictates what the cell is supposed to do. Okay? Okay. In a hypotonic solution, the ratio is low solute, high water, or high solvent. So in an animal cell, if there is too much water, it would lyse, or it would swell, it would burst. For a plant cell, it would be turgid, in other words, normal. So yeah, basically explode. Other unique structures or organelles, we have cilia and flagella. The only human cell with a flagellum is the sperm cell. That's it. Passive transport. Molecules are moved within and across the cell membrane, therefore transporting it along a concentration gradient without ATP. Alright, let's just skip this part. This table may be useful. Why did I crop this picture like this? I remember. There's a lot more information than just what I cropped. But these are the most relevant ones. Let's check the complete reference. Okay. So, active transport is at the left. Okay. So for active transport, transportation is only in one direction. That's for active. Active transport is highly selective. It is influenced by temperature, and active transportation is influenced also by metabolic inhibitors. For passive transportation is bidirectional. I suppose it is implied here that, for example, in breathing, you inhale and exhale nonpolar molecules such as oxygen and carbon dioxide. When you inhale, the cells receive oxygen, and when you exhale, the cells release carbon dioxide. So transportation is bidirectional. Breathing is associated with just passive transport. there is no need for ATP for the process to ensue. So passive transport is partly non-selective, and it is not influenced by temperature as well as by metabolic inhibitors. Okay. Circulation. Circulation is from a region of high concentration to low. The process, it takes time, of course. You don't need ATP, but the con is passive transport takes time. More time than if you involve the use of active transport. So what is the aim of passive transport? To transport all soluble molecules. So two of them I've already stated earlier, oxygen and carbon dioxide for breathing, and for inhale exhale. As well as water, really. Water is polar, so how can it undergo passive transport? Maybe it does make sense for water to be eligible for undergoing passive transport. It's glucose that is nonpolar. Let me just confirm that information. Really? Glucose is polar. But for some reason, glucose can only be transported via active transport, not passive. Maybe whether it's polar or non-polar, it cannot be a basis as to whether they can be transported passively or actively. Anyways, lipids, sex hormones. Again, this is associated with steroids, estradiol and testosterone, among other things. For the protein, carrier proteins are not required. Again, there are just three types: simple, osmosis, and facilitated. Major function of membrane proteins, there are 6: transport, signal transduction, cell-to-cell recognition, intercellular joining, enzymatic activity, and attachment. Cell membrane is, in other words, the functional mosaic. Okay, so let's talk about the active transport this time. So enables a cell to maintain internal concentrations of small solutes that differ from concentrations in its environment. So what you're doing, it requires energy because you are going against the concentration gradient. So molecules are moved across cell membrane, being pumped against the concentration gradient using ATP, or cellular energy. So for the circulation, there's a movement from a region of low concentration to a region of high concentration. The process, it takes it's really fast, basically, and the aim is to transport all polar molecules, basically proteins, large cells, complex sugars, ions, water, etc. Okay, so water is also mentioned here. Let's confirm this information. Can water be transported by passive transport? Okay. How about active? Okay. So it turns out that water can undergo active or passive transport. Okay. For the proteins, carrier proteins for sugar are required. So what are the types? Under active transport, we have exocytosis, endocytosis, sodium-potassium pump, and proton pump. Okay. ER. I've mentioned this earlier. Smooth ER, synthesis of lipids, specifically cholesterol and phospholipids. They form the membrane of the cell. So without the smooth ER, the formation of the cell membrane, particularly the phospholipid bilayer, may not be complete. For the rough ER, this is where ribosomes attach. And yeah, there they synthesize proteins. Some other ribosomes that are free floating in the cytoplasm, uh, they still make proteins, I suppose. But yeah, it just so happened that certain ribosomes stick to the rough ER and do the same job. Proteins and other materials emerge from the ER in small vesicles. Okay. Okay, what is the general definition of the ER? It is a membrane enclosed passageway for transporting materials. Okay. Two broad categories of cells. You have eukaryotic and prokaryotic. For eukaryotic cells, they have nucleus and membrane enclosed organelles. They are multicellular, and we have these in plant and animal cells. For prokaryotic cells, they have no nucleus and membrane enclosed organelles. They are unicellular. One example is bacteria. How about fungi? Is fungi? I'm pretty sure it's eukaryotic. Yeah, fungi are eukaryotic. Okay. The plant cell would be plasmolysed in a hypertonic environment. Animal cells will be shriveled in a hypertonic environment. Animal cells will be normal in an isotonic environment. Plant cells will be flaccid in an isotonic environment. Plant cells will be turgid in a hypotonic environment. Okay. Animal cells will be lysed, or they will burst in a hypotonic environment. Okay. Finally, we're finished. That's it for this flashcard deck. I'll make more soon. Thanks for watching.